Here. And I have the absolute pleasure to introduce and welcome to our stage here Dr. Bert de Vries and Jolijn Verseput. And he's known as Bert and she's known as Jolijn here in America. So they came all the way from the Netherlands, as you know, to join us here, to be the experts, to answer all your guys' questions, and to help us with getting on the same page with the research we're doing. They're going to give you a lot more information on how you can help get involved in that and what is happening and what do we know. So without further ado, Dr. Bert and Jolijn.
It's wonderful to be here. I'm impressed. It's always nice to meet kids with JdVS. I'm impressed by the siblings. I'm impressed by the parents and all the other family members. And I'm impressed by the organization. So Kara and team, well done. Okay, so yeah, let me see. Yeah, so first I had this title, Getting to the Janssen and Vries Syndrome, the history and a glimpse of the future. Kara and team gave me a very nice title, or us. It will be a dual presentation, so I will start and Jolijn will take over. News from Radboud, I think that's very lovely. By the way, so when we're talking about Radboud, it's the Radboud University, and that's located in Nijmegen. It's the oldest town in the Netherlands. So it has been founded by the Romans more than 2,000 years ago. So it's a wonderful city to be in. And as you can see, it's a little bit at the eastern part of the Netherlands. The Netherlands is 200 miles from north to south and 100 miles from east to west. So if you do this, yes, it fits in Illinois very, very lovely. So then you know where we are coming from. It's about history, and I always like to start with real history. I've done this before, so Antonio van Leeuwenhoek, he is a Dutchman as well, but he grew up in Delft, so that's a different town, also very old. The thing is, Antonio van Leeuwenhoek did something impressive. He developed a microscope. And he was very good in this. He was one of the best in his time. And he was curious as well, so he started looking at all kinds of tissues. And he also looked at a certain moment in time in sperm. To be honest, the story is it's his own sperm, by the way. So he started looking, and he saw the sperm cell, and he saw the tail, and he saw this head. And then he and his colleagues thought, okay, so what's in the head there? And they thought, okay, there might be a very tiny, tiny, tiny little human being there, and then transform from the man to the woman and the fruitful womb, and then grow up to a lovely baby boy or girl. Interesting thought. We are talking about the 17th century. So we learned more. So nowadays, since the 50s, we are familiar with the chromosomes. And chromosomes are the carrier of the genes in each cell, and if you look at them with a better microscope, of course, then you can see that in each cell there are 23 pairs of chromosomes. They are easily numbered, and then the 23rd are the sex chromosomes. If you have two X chromosomes, you're female, and an X and a Y, you're male. Like this. So anyway, but if you look into the chromosome for children with Janssen and Vries syndrome, then actually you won't see any difference between normal or regular people. So we went next step. So now I skip 50 years, and starting in 2000, we developed novel technique. So that's one of the DNA chips to look more accurate at the chromosomes. So your resolution becomes better and better. But still, this was a huge step in 2000, and we pioneered this as well in Nijmegen as one of the first, but it was not good enough to identify or to find the gene for what has been renamed later on JdVS. The real step was when we get to exome sequencing. And exome sequencing means that you test all the genes in one go. And that was really a revolution. Maybe you have heard about this. Next generation sequencing, it's called. And here again, we start pioneering this. The principle is, from the outside, relatively simple. You have your DNA, you put it into a machine, and then the sequences are coming out at the other end. That's very simplified, by the way. But then you get lots of variants in all different kinds of genes. And then the very important question is, which of all those novel variants are actually clinically relevant, linked to a disorder? And here again, we were in Nijmegen very well, started pioneering this very well as well. To be honest, we started off with doing what we called a trio analysis. That means that we tested the child and both parents in one go, and see whether there were any changes coming from the parents towards the child with a neurodevelopmental disorder. And we started off with 10 trios. Why only 10? Because it was highly, highly expensive. It cost us $100,000, euros, dollars at that time, to do these 10. But we did it, and we were very successful because in those 10 we were able to make a diagnosis, a novel diagnosis in 7. So that's a diagnostic yield of 72%, and that's pretty high. Okay. This girl was referred to my clinic in 2015. She's the first girl where we, at the end, identified Jensen-de-Vries syndrome. It started off with, so she was 5 years old, and she was referred because she had a delayed in development. She had a normal birth weight, normal pregnancy, but a little bit lower birth weight, I must say. But she has this feeding problems. She had a reflux, and she has hypertonia, that's a low muscle tone. Her IQ was estimated as being 72. She had problems in concentrating. She had a little bit special behavior, but she was very open in communication. Okay, very recognizable. She had a high pain threshold and was relatively sensitive for sound. This was what I recorded in the file. And that's what we do. You do a physical examination, and now she was a little bit short, but she had a normal head circumference. And then I'm also, I'm a clinical geneticist, and that's also dysmorphologist. Then you start describing what you see. And that's far more detailed. We have an eye for this, so deep-set eyes, a little bit upslanting palpebral fissures, a short upturned nose, short filter and broad mouth, and a little bit low-set ears. And if she walked a little bit unstable, broad-based. Okay, I didn't have any clue what the diagnosis was at that time. But what we did do is that trio analysis. And in those trio analysis, we identified this de novo mutation in PP1-ND. Okay, one girl, 5 years old, with a de novo mutation in a gene. I hadn't heard about this gene before. So what do you do? So we already had some experience with over 800 children that had been tested, and I requested the lab, go back to all your data and look whether you find similar children with mutations in the same gene. And lo and behold, there were 2 others. And I still remember the moment that I got the photos in, and I looked at them and put them next to each other, and then I called Sandra Jansen in. She was a PhD student at that time, and said, Sandra, look, maybe we are looking here at a novel syndrome. Based on these 3, we thought, okay. So that was really what you call a eureka moment, as you can see here. Okay, so they look alike. They all 3 have difficulties in the development. So what does PP1D do? It encodes for a phosphatase, and it's involved, and this protein is involved in DNA damage response pathway and linked to P53. Okay, how does the gene look like? The gene has 6 parts. That's called exons, and all the I must say first of all we went out there and we looked whether there were other patients around the world. We have a network, and we were able to collect 14 other children, or the complete, so 11 other children. So we had, at that moment, we had 14 what we call patients as clinicians, and they all had these mutations at a specific spot. So again, so if you look at the gene, it consists of 6 parts, and they are called exons, and the mutations were all in exon 5, at the end of exon 5, at the beginning of exon 6, as you can see here. And that meant that there was a shorter protein, what we call a truncated protein. So there was a protein formed, and in that protein, the phosphatase activity, and that's here in red, was maintained, but the nuclear localization signal was lost. That's here in green. So they are at a special part. So there was a sort of what we call sort of gain of function, probably, through the extra phosphatase activity. Okay, so then we had those 14 patients, and then you ask yourself, okay, it's lovely that you think about 3 might be a novel syndrome, but does the others also look like the first 3? And lo and behold, yes, it was. They all have a broad forehead, low-set ears, and what's very special is the nose, but also the mouth. And the mouth is a little bit broad and with a relatively thin upper lip. And if you look at the hands and feet, then they turn out to be relatively short and broad for those where we had data on. Yes, so this is a novel syndrome. Then we start looking at other features, and then that's what you put in the table. So all but one had the intellectual disability. The hypertonia was in the majority of cases, so low muscle tone, especially at young age. The unstable gait, approximately half. And the sensitivity of sounds, that was already recorded in the first 5-year-old girl. You could see it frequently. It's just a common thing. And also some behavioral problems. And then the other thing is that there were some problems with gastrointestinal problems, the feeding difficulties, but also the reflux and the vomiting was present in the majority of cases, and also the constipation. So this is all based on the first series. They were relatively short, not all, but the small hands and feet on which we had data all had small hands and feet, and some developed a sort of extra curvature in the spine, what's called an hyperlordosis. But there were also some other issues, so periods of illnesses. The high pain threshold was also reported in this 5-year-old girl. We saw frequently some vision problems, hypoplastic nails, and recurrent infections. Okay, so 2015 we saw the first girl. In 2017 we published this in the American Journal of Human Genetics. And there Sandra Janssen is the first author. She was a PhD student of the project, and I was leading the project. And after this publication, this syndrome was renamed Janssen-de Vries syndrome by official organs, so like OMIN and so on. So that is the name coming from. Okay, but then it was only 14 cases, so you want to know more. You want to know the whole clinical spectrum. And you also want to know natural history, so how things evolve in each child. And here Cynthia Curry is coming in, and she will be presenting her lovely data later on. She's sitting over there, because she was able to connect with 40 researchers from all over the world and start collecting data. And she will present the details later on. And she was able to collect 37 individuals from 34 families. And that was published, and the publication is on your desk, I guess, so if you want to do some good reading, please start doing this. So there are lots of co-authors, and this was the first step to get into the clinical spectrum. So what can you expect and what can you not expect? And that's a huge achievement. And of course, we are starting interesting in the natural history. And for the natural history, we're starting 2 projects, and now I hand over the presentation to Jolijn.
Hi everyone, it's lovely to be here. So now we've heard a lot about how everything evolved for JdVS up till now, but I don't have to tell you all that there's still a lot of information that we're missing. So that's where I come in, partly of course. So nice to meet you all. I'm Jolijn, or Jolijn if you would pronounce it in Dutch. I'm a medical doctor and now PhD student at the Radboud UMC under guidance of Bert. And I'm working on 2 different projects which will involve JdVS as well. So the first one is a natural history project that I will be starting with, telling you a bit more about what it is and also some very, very preliminary results, because some of the families maybe also here present today are already involved in this project. So I have some very first hot off the press results of the project so far. And of course, if you were here 2 years ago, and also you've heard a lot about this already, we're doing a 3D imaging to learn more about the faces of children with JdVS, which I will also be sharing some first results about today. So is this for, yeah. Okay. So before I can get into the natural history study and what it is, I first need to introduce to you the platform that we're using to upload the information, which is the Human Disease Gene website series. Maybe you've been to our BPM1D page before. If not, go check it out. But this is a platform that we developed at the Radboud UMC where for many, many genes, people from all over the world that are experts in their own gene field can what we call adopt a gene to say, okay, I'm knowledgeable about this gene, about the disorder that is linked to it, and they can share their knowledge with us, but also collect new information. And that new information used to be coming all from other clinicians, other researchers that had a new individual that was diagnosed with a specific disorder. And that was going quite well. So for Vries, we have about 16 individuals that have been clinically included, for which you can see a nice graph and chart about all the health issues, developmental issues that the clinicians shared with us. But if you have a clinician, they will only have a very limited amount of information about the individual, because they only will see them once, maybe a few times at one point in their life, and then their information stops. But you as families have, of course, much more information about the individual that you're caring for. So we thought, how about getting you guys as families involved, because that will give us so much more information to learn from. So that's why we started the Natural History Project. What is a natural history study? It's a study design that's used moreover to learn more about the natural course of a disease. So we as researchers don't tell you to do anything. We just ask you lead the normal life of your individual with JdVS. Of course, if they need treatment, go get the treatment that your doctor prescribes you. But we don't say you need to take this drug or you need to do this treatment. We just want to know if we as researchers don't do anything, what happens. So to learn from you about this, we launched a new, what we call patient portal, which has 2 questionnaires. One about development and health, and one more about practical and social skills that your individual with JdVS has developed. And if you get involved in this project, you will get a request to yearly update your information so that we can, over time, learn more about the JdVS individuals. Well, of course, I don't need to tell you why this is important, because that's why we're here today. We want to know more. There's still a lot of information lacking, especially what we call longitudinal data. So if you have one child with JdVS of 7-year-old, you cannot compare them to another one of 8 years old to see what will happen in the next year, because everyone is unique. So to get data on how people develop over time, you need to follow individuals over time. So that's what we will be doing. And we've heard a lot about our beautiful adult individuals, present here also in the room sometimes. That's also where a lot more information is needed to learn for parents that have a diagnosis at a young age what will happen in the future. And then as a next step, which might be a bit further away, but at some point we as researchers would like to help you with what kind of treatments are needed, do specific testing for specific interventions that might be more helpful for JdVS individuals. If we want to do that, we first need to know what is normal for JdVS individuals so that we can learn what impact does it make if we give them specific treatments. So if you want to get involved, you go to our BPM One Day page. I also have some QR codes that are in your bags, I think. Kara has been graciously providing that, and they're also on the back of your folder. So don't do it now. Just take a moment later maybe if you want to get involved. But there we have a page where you can find some more information about the study than I can tell you right now. And also sign up as a family to start enrolling in this study. If you do so, we will create a portal for you where you can find your questionnaires. That will have a lot of, we've tried to make it as easy as possible, so it will be a lot of yes-no questions about development, about neurological features, organs like your lungs, kidneys, hearts, to learn what are the problems that your JdVS individual is facing at this point in time. So I've got a question before, do I need to fill it out for the past life? No, we want to know what's happening now, and then next year we can see what has changed over the past year. You fill it in, at the end you will see a pop-up. If you also want to fill in the second questionnaire about the daily and social living skills. If not, you can also just close it, or if you want to, you can fill out the rest of the questionnaire. From some people that have tested it, I've heard it takes about 30 minutes to an hour to fill it in. And then after a year, you will be asked to start completing the rest, where we are aiming to make sure that all your previous answers are saved, so you only need to change what has changed over the year. So the next time it will be much shorter to fill it in. So we're hoping to getting you all involved. So these are the QR codes. They're also in your bag, so don't stress about scanning them all now. If you have any questions about this project, please come find me. There's also a QR code for a webinar I did with the JdVS Foundation that has much more information all about the consent and what you're consenting for if you're getting started. Oh, and I was a bit dumb because I forgot one very important part to put in these slides, but I want to tell you. We're also aiming to get a graph and chart up for the family enter data. So there's already one up for the clinical enter data, but we want to get one up for the family enter data as well, so that you as a community can just, at the real time that people are adding information, learn from each other. So that it's not just us researchers having all the data and you don't know what's in there, but you can also learn from all the other families that are sharing their knowledge. But that's something we're working on very hard right now. So that's natural history study, because we want to learn more about what we do. But like I said, we already have some individuals that are already participating. So we have, for now, 28 families that have signed up, for which 16 have completed their forms already, which individuals for 6 different countries, mainly the US, but also someone from Italy, UK. So word has gotten out there that we're doing this, so it's great. Ages 1 to 19, and for now, 11 boys, 7 girls. So I wanted to share some very, this is only 16 individuals, right? So we cannot really give you all the information because we still don't have it yet. But I wanted to show you the developmental milestones that have been put in so far. And of course, 2 very important things that you've already been talking about a lot, behavior and the gut issues for the data that we have for now. So for development, we ask about ages of sitting, first steps, first words, and more about intellectual disability and IQ scores. So for the individuals that are in our study right now, ages of sitting were between 6 and 12 months. Ages of first steps between 16 and 30 months. First words, 9 to 24 months. And IQ scores range for the individuals present in the study now from a mild intellectual disability to even normal IQ scores. For behavior, we have many questions that ask about what kind of behaviors could be present. And keep in mind that this is a standardized questionnaire that is rolling out to other syndromes as well. So you might miss some specific JdVS questions at this moment, which we could add in the future, but for now it's standardized. So we always ask about ADHD and autism, that were present in about a quarter to a third of the individuals, and then many scored yes on things that I think will sound familiar, like the anxiety, of course, short attention span, impulsivity or restlessness, and a minority also said that obsessive behavior could be a thing, or sometimes even aggressive behavior towards themselves or other individuals. But which I was pretty happy about for you as families, that sleep was usually scored to be quite normal. So even if your individual could be challenging over the day, at least I think many get quite a good night of sleep in. So I think that's very positive. And then lastly, of course, gut health was also something that was popping up all over the place for all of these individuals, with constipation present in about 88%, feeding difficulties and easy choking being very often present for these individuals. Reflux, so bile coming up from the stomach, also being present quite often. And hearing from you all today, the cyclic vomiting, I think we need to do some more in-depth questions about that at a later point, because this 90%, I don't think that covers what really is happening. So there we need to ask you maybe some more later. So that's our natural history up till now, starting from May until, well, this month. So if you're interested in participating, please sign up. Keep in mind that I need to do some things behind the scenes to get you started, so I'm here now, so don't get worried if you don't receive an immediate email that you can get started. I'm working on it, will coming quite soon, because this is very much to be continued, and maybe in 2 years' time, if I'm here again, I can share some much better results. So that's the first project. Then let's go into the second study that I'm working on, which is a 3D facial analysis study. As clinicians, especially for children with neurodevelopmental disorders, looking at the face and other features of the body can give very important clues on what is happening or what diagnosis could be present for the child or the adult that we're seeing in clinic. So the way that the shape of the eyes, the nose, the ears can tell us more about the potential diagnosis. And if you look at our JdVS individuals, like Dr. Vries already put up, we know that none of them look exactly alike, of course. But if you look closely, and Dr. Vries also talked about this from the frontal images, then the mouth very often can give a clue that JdVS could be present in an individual, or the forehead, broad forehead. However, these are not that many clues for clinicians to make a diagnosis upon. So we were wondering what more can we learn about the faces of our JdVS individuals that can help clinicians in the future to make a better diagnosis. So these results are all from 2D imaging, so we thought maybe a good step could be going into 3D imaging. So that's why 2 years ago we started this project, and we're still continuing it now. If you want to go from a 2D image to your 3D facial analysis, I put up my own head because why not? If you take a 2D image and then go to 3D, this is always very confrontational. Please don't look at your own 3D images if I make them later. I know, it's awful. But we do gain a lot of information. But because from those very flat 2D image, we've now moved to a 3D image, which has a lot more information about the depth that is present in our faces and gives more information about the shape of the face. However, if I want to analyze these images, I make these kind of 3D masks with a technology called MeshMonk. And then it's good to keep in mind that we also lose some information in the analysis, because this technology just cannot grasp, for example, the intricate shape of our ears. It's just too hard for now for the current technologies to analyze that. And you also lose a lot of facial hair. But if you make these kind of MeshMonks for all your separate individuals, you can do very nice analysis with them because they're standardized and you can easily compare them to other images. So that's what we did for our first analysis, which we call a mean signature analysis, to get the signature image of our JdVS individuals. So at the last conference, we collected many images, of which 9 were of good enough quality to use for this analysis. And what we then do is we take the shape of our individual face and compare that to someone with the same sex and the same age to see what is different in the JdVS image from our control images with someone with the same age, the same sex. If you do that for your individuals, you will end up with what we call some kind of a heat map that shows in the face what are the regions that are different from the control images. If something in the face is more broad, more going outwards, that will show up in the warmer tones. So orange, red means that the part of the face is more going outwards, whereas things that are going more inwards or more sunken into the face are shown up in the cooler tones. So here you can appreciate that this individual has round cheeks, which show up in bright red, whereas the more narrow nose of this individual and the small mouth, they show up in blue. So we did that for all our individuals with JdVS and then took a mean out of that to see what is the mean signature, at least for these 9 individuals. So just a quick recap, we would expect something to show up around the mouth, maybe the forehead. So let's see what happened. This was the mean signature that came out. So I think the mouth very nicely shows up as we would expect. So the broad shape of the mouth, the more prominent upper lip that we see in many JdVS individuals also is very nicely present in our mean signature. But we were also very surprised to see a potential new feature for JdVS, which we called supraorbital grooves, which is just a fancy way of saying there are these small grooves or indents above the eyebrows. So we saw that more expressed in some of the individual images, but also in our mean. They're showing up nicely symmetrical, which gives us a clue that it's really a thing. So that could be a potential new feature for clinicians to help get the right diagnosis. And I'm very curious to hear from you as families if you recognize this from your own individuals, and to see what happens if we would get a bigger group, if it's still a feature that remains upright or keeps standing. But this is only a mean, and as researchers we're always very curious of a mean because a mean can leave out the outliers. So if you do an analysis with a mean, then you also need to check how consistent are the features that are showing up in your mean image. So that's why we did follow-up to check the consistency, which is called a cosine distance analysis. Please forget that right away. But that's what it's called. And there we use actually the basic mathematical principle of a cosine that helps you calculate angles in a much more fancy way to see how far away are the individuals from each other if you put them in a 3D space. So actually what we just measure is how similar are these images to each other. And if you do that with this kind of analysis, you will end up with a value that is between 0 and 1. If your value out of this analysis is exactly 0, that means that all your individual faces are exactly the same. If your value is exactly 1, it means that they're all over the place, there's no clear relationship between them. So for a syndrome like JdVS, where we know that there are similarities, you would expect your value to be at least under 0 and the closer, or under 1, sorry, and the closer to 0 it is, the more consistent your faces are compared to each other. So we did that for our JdVS images and also compared that to 21 other neurodevelopmental syndromes that we know have good consistency in their facial features. So that leaves us with a very long list of bar graphs where you might see that a very consistent syndrome like Down syndrome that is easily recognizable for many people because their facial features are quite consistent. Their value, of course, also is not 0 because none of them look exactly alike, but they have a value of about 0.5, meaning that that's a value that shows good consistency. And at the bottom you see unaffected, unrelated, meaning that those are the controls. So they're individuals that have no relationship to each other, not the same syndrome, not family related. Our JdVS individuals ended up actually out of this list, the closest to the controls, meaning that at least out of these list of syndromes, our individuals were the least consistent. That doesn't mean that their features are not consistent, it just means that out of this list, they are the least consistent, which we could get a little bit because we know that there are many differences in many parts of the face, but we were wondering what about the mouth and what about the forehead, because there we know that we would expect more consistency. So we did a last follow-up where we divided the face into 8 different regions to see, okay, this is a total, that's not that consistent, but how about the different regions where we might expect more consistency? So that ended up with a graph that looks like this, where for our total face and many of the facial parts, we know that the consistency is at least as inconsistent or sometimes a bit more inconsistent as the total face. However, our lip and our forehead regions where we would expect more consistency do indeed show that. So the lip goes to about 0.6, 0.5, which means that the lip area is very consistent for our individuals as well as the forehead region, at least showing a much better consistency compared to our total face. So that's some first results that I could get from the first images that were taken 2 years ago, but I don't think we're already at the end of what we could learn from these images, especially if we got some more. So that's why I brought a new 3D camera again this year, which has even better quality of images, and there's also many new individuals present here today. So we're very happy that we can make new images if you as families are still interested. You have a little folder in your map or your binder that tells you more about the 3D study that we're doing, also the consent you will be giving. So if you are interested in participating as a family, please come find me at the boardroom right across the hall, boardroom East. That's where we'll be setting up during lunch. So if you have some spare time, if your individual, your champion is with you, please come find me over there all afternoon. So we can take images of your champion, but we like to include family as well because you are of course also passing on some of your genetic information about your face to your individual. So we like to see how you compare to your individuals. So whole families are very welcome at the boardroom, so we can keep building on this data and keep learning more and maybe do something with AI, fancy words, to even get more information about the face in the future. So that's all I wanted to tell, and then I think Bert has some closing remarks for you.
Yes, so whole family, siblings, parents, uncles, everyone come over, because then we can learn. And there's a reason behind this. It's not only about making the diagnosis easier, but the development of the face is starting from the same cells that form the brain. So we try to make sort of links at a certain moment in time, but for this we need the data. And similar, and that's what I also told 2 years ago, you have the knowledge, we can only collect. So you have to share the knowledge with your child and the family. So that's where we're focusing on. So glimpse of the future, and to be honest, the future is ready now. And one of the future things, or the things we are currently doing, and especially also doing this in Nijmegen, we can change stem cells into neurons. That's called eye neurons, induced neurons. And the interesting thing is that if you do this right, and that's what we do of course, then they form a network. You can play them. And the interesting thing is, those neurons, nerve cells, they start communicating with each other. It's impressive. You have to treat them well, but they start communicating with each other, and you can measure this communication. And that's done by a multi-electrode array that measure the interaction of those nerve cells or neurons. And let's see. Oh, now can I? Oh, what? Oh yeah, at the back you have to press somewhere. I don't think I can do this, because what you see here, and I cannot point it out because there's no pointer, you see all the electrodes. Oh yes, there it starts. I'm always impressed. So what you do see here is, when you measure they have spikes and bursts, and they start communicating with each other, and this is how it looks. You can measure this, and what we experience is that it's for different neurodevelopmental disorders, the pattern is different. So the way they communicate with each other. And this is very special, because this is how you can define how neurons communicate with each other for a specific disorder. And this will also be the starting point of correcting things, because we do know how neurons communicate in a normal situation. Okay, so this is, we can look for hours. It's like a thunderstorm. That's 2 days ago, sort of. This is important. These are our next steps. What's even more, not more important, similar important, is that we can grow the cells into what we call brain organoids, so mini brains. That's what we are doing. It's tedious work. It costs lots of money, but we can do it. And then we can study, we know how the normal situation is, and we can study the development in early stages, for instance, for JdVS condition. So, very important. So, we can already do this if we have the right financial support. Okay, that's what I briefly want to bring in, sort of a glimpse of the future, and we are open for questions, because that's why we are here, to address your questions. So, thank you. Are there any questions? Is someone walking? Oh, there's. Oh, there was one up from.
Will we have access to this presentation in any capacity?
Sorry?
Will we be able to get this presentation at all?
I think it's recorded. Oh yes, and thanks to all the families and individuals, by the way.
I actually have too many questions, so I'll try to keep it to just a couple. But have you found any differences in symptoms between the individuals with our syndrome, whether they have the mutation on exon 5 versus exon 6?
Good question. As far as we know, there is no difference, because the underlying principle is similar. The thing is that you get this shorter protein, and one of the signals is lost, and that the protein is transferred from the cytosol to the nucleus, and that's the nuclear localization signal. If that's lost, then we think, but we have to measure this, we already know, because that's also in the original publication, that it's normally formed, but we think that it will stay longer in the cytosol, and by doing this, it's less quickly degraded. So that's the idea, and that's why we think it's sort of gain of function, and that's the phosphatase activity is retained a little bit longer, and we think that's all, that's what we think, but we have to do further research on this, so, yeah.
One other question I have is I had read an article about an individual who had mosaic JdVS. Does that develop also de novo, or would that be something that they potentially had a parent with it, but now it's only present in a certain part of their body that's studied?
Yeah, so mosaicism means that in part of the body cells you have the mutation that can occur indeed during pregnancy or later on. It's not very common as we see, but we don't know whether, because then you have to test lots of things. So what we we test routinely, or in the Netherlands and also in other parts of the world, in blood. And sometimes you see in blood that it's not 50-50 in blood, and then it's a little bit lower in blood, and then you think, okay, might this be a moisetism? But most of the children with moisetism, they are unique, not only for in a case that it's difficult to compare all the children with moisetism, and we don't have large numbers anyway at the moment. But that is a common thing, and maybe, and that's one of the things why we're also interested in the natural history, because if you start Googling this gene, then as a parent you will be shocked, because it's doing lots of things later on in life, and we don't have any evidence for this in children with JdVS, but it might be related to cancer. And that seems to be a worry, might be a worry, but so far we don't have any evidence, so probably that's is a different mechanism.
All right, before we take another question, I'm sorry, real quick. Anybody with children in childcare, it's 12 o'clock and lunch is about to start, and we need to go ahead and check them out. I didn't mean to interrupt.
Yeah, but fair enough.
We can take a couple more questions, but I just wanted to let people know.
The 3D study that you guys are doing and gathering information on, will there be how does that information then get to other clinicians and folks that are in the diagnostic process? Because I think the goal of that is to help with diagnostics, right? So, and maybe just to understand, but diagnostic primarily, right? For people we don't know currently have it. So is there a deliverable that's gonna, or how do clinicians get access to that?
Yeah, so for the natural history, like I said, we're working on our graph and chart. So all the information that will be in like the back of the website where you have entered your information, we're working on getting that up on the page as well, so anonymously, so no information will be retraceable to your champion, but other individuals could definitely see it, as well as the families, but also of course the clinicians, so it's just open information up on our website. So you as families can just easily take it all down, present it to your clinician saying this is what's present. For the 3D, it will be a publication that's coming out.
Yeah, so I can a little bit stress the importance of the Human Disease Gene website for PPOND, because we will try to keep this up to date with all novel information. And as Jolijn is mentioning, you can print it and just have a look at the graph and chart and see what's already on there, and you can actually print this out and go to your clinician and say, this is JdVS, these are the features you might encounter, so please check those things. And we had just have a brief meeting, and Jay will putting some information on. He's working on a mouse model for JdVS, and he will put some information there. Cynthia is much involved all the time with the website, so she will also put information there. And Dr. Lee, I don't know, oh yeah, he's there. He will put information there on his experience with CVS and what you can actually do to make these things work better.
For the yearly update, is that something that you guys will reach out and kind of give a reminder for?
Yeah, okay. So we'll reach out. You will get a request to enter your new year of experiences. So like I said, we're working on making sure that everything like a birth weight and the first steps, if your individual is past that age, you don't need to upload it all again and again and again, because it won't change, obviously. The things over the year that have changed, you can tell us so that we know what has changed and we can keep building on what is changing yearly for your champion.
And again, I stress the importance of this, because you have the data, we can only collect it. So please start doing this. It's just in our work. It's not too much, I guess. Maybe,
Of particular? Of particular what? Sorry.
Maybe you...
Oh, well, so far, so the question is if so far at Natural History we found any specific symptoms for people from a particular descent. So far, there's only individuals with a northern white descent in the questionnaire yet. So there's no way to tell yet.
But we are also interested in that, and
Yes.
the website will not stop. It will be there in 30 years' time. Still, it will be around. I stop by then, but keep this in mind. So this is not a short. We are not in there for short-term projects. We are in there for long-term projects, and that only will allow us to answer your questions. And I was impressed by the 4 ladies who were sitting here this morning, showing what adults with JdVS for their issues. But we can build this up in large numbers. And that will be very structured and a little bit more scientific than anecdotal. So that's the idea behind the whole thing.
I was asked yesterday by someone, so for how long will you be doing this? So if it's up to us, 70 years, even longer, because that's the lifespan that we now heard about, 77. So if we really want to get all the data, we should cover the entire lifespan. So we'll see how it goes. But if it's up to us, we'll just keep going until we know everything that's there is to know.
Use the mic.
One last final question I had. I know when you were talking about kind of the similarities of different things with feeding, all the other things, and you discussed mildly decreased to potentially normal IQ scores so far found. Has there ever been anybody that's been found to have a higher than normal IQ score within the syndrome?
Someone, you want to answer the question? Yeah, so that's also what we'll start collecting and show what the variability is between different. So we are all unique. So all JdVS champions are also unique, but then still we want to bring this in a more structured way to show what we know. So, any other questions? I think we're all in for lunch, I guess. Okay.
Of course, if you have any questions, please come find us during the next 2 days.
Yes, so lunch will be upstairs in the